So today our topic's about the cell structure and function, which is essentially the starting topic for most MCAT lectures, but we're using the syllabus guidelines from topic 4 to essentially guide us through the topic. So something that's important to understand here is that the learning objectives will range from the structure of the cell itself. For example, the cell wall, the organelles within the cell, and then eventually compare two kinds of cells, which are the prokaryotes and the eukaryotes. Now, I understand that because this is a MCAT or MDCAT or NMDCAT lecture, essentially, so every student has a basic idea of what's going on, what structures are being used. So my purpose here is going to make sure that you understand proper functioning of each structure, and then you understand why each structure is important and the way it will be tested. So it's less of a understanding what a cell wall is, but a more understanding which organisms have what cell wall and being able to answer their questions essentially. So we'll use a very basic three-dimensional diagram of the cell. I really like this diagram because it shows you the cell as a floating body, right? Which essentially it is, unless you make it non-motile. So there are just a few things that are very basic, is understanding that you have something called a nucleus. Now you can see that it's the nucleus, it's definitely an animal cell, and you know that since you were a kid because you've studied that animal cells only have cell membranes, no cell walls. Animal cells have a central nucleus, no large vacuole. So all of that comes true when you look at this cell. Essentially, you can see that it's floating in a cell cytoplasm, which is why this diagram is so amazing. And then you can see within the nucleus you have a nucleolus. So we're going to study why I keep calling this the nucleus. Then we're going to study these little structures such as ribosomes, or the mitochondria that is there, and it has a specific structure, or vacuoles, or for instance, let's say this deposition of double membranes and how it's different from the one surrounding the nucleus. So if I say this purple covering is for the nucleus, which has the nucleolus inside, it's wrapped by a double membrane deposited on it. So what is this? Obviously, if you remember well enough, it's the endoplasmic reticulum. My job here is to make you understand the differences here and what exactly is happening. And lastly, to understand, when you look at the cell, you should be able to tell that, okay, certain things work certain way. For example, you look at the cytoskeleton and you understand for these threads are shown as the cytoskeleton. So you know, obviously, that the cytoskeleton is responsible for, let's say, transport and maintenance of shape, etc. And you can also read that out in a book, but what are things that you should be able to read from a diagram and be like, okay, this is what the cytoskeleton is doing. And so if I have a good concept of the cytoskeleton, the moment I look at this diagram, I understand that it has hook-like structures here and hook-like anchors here, from which these green cytoskeleton filaments are anchoring out. So that gives me an idea, okay, if there's a second cell just like this one placed over here, it must be attached through this area. So this cytoskeleton is helping attach the cell to the surrounding tissue, or to give the cell its own shape, right? So these are stuff that you need to understand right as you jump into the diagram. So let's move on and see all of these in detail and try to understand them. Most of my slides are pictorial. There's very few notes, and most notes should be made as I'm speaking. Make sure you take images or screenshot these and make notes next to these as I'm explaining. So essentially, the organelles that we need to talk about in detail are the chloroplast, because we're also talking about the plant cell. Right now, remember, I'm generalizing the cell and their organelles. I'm not specifying which one's which. So we're talking about the chloroplast, we're talking about the Golgi, and the endoplasmic reticulum, and then obviously the nucleus itself, a lysosome, and a mitochondria. And like I said, the best thing that I can do for this topic that you prepare, that you've been doing since I'm sure you were a little kid, is when you look at this diagram, you understand what this diagram is saying. So right now it's just lines and folds, but from this slide onwards, it's no longer lines and folds, right? So if you look at so that was 3D diagrams. This is a one section, one diagram in which you can look at the cell with the cell, cell membrane, and there's organelles inside. So to quick judgment, you should be able to understand that that this is the nucleus, with the dot inside obviously being a nucleolus. Now we need to understand what that structure is, right? And then followed by smaller worm-shaped bodies known as mitochondria. So that's clear. And if it's a plant cell, so that's an animal cell, right? If it's a plant cell, then mitochondria are present, but other worm-like bodies, such as the chloroplast right over here, and these are also, look at them, it's also cylindrical in shape, right? That must be present somewhere here as well. And then there's a deposition of membranes in a corner called the Golgi apparatus, and a deposition of membranes, double membranes around the nucleus called the endoplasmic reticulum. So let's appreciate these here as well. It's just a deposition of membrane. Imagine it's like dough, it's like pizza dough. So you fold one layer on top of the other, on top of the other, or it's like a naan, you're folding one layer on top of the other. If it's surrounding the nucleus, it's the endoplasmic reticulum. If it's in a corner sitting there, look at this, in the corner sitting there, it's the Golgi apparatus. So being able to identify it is very important. Now moving on to the nucleus, what exactly is the nucleus? So the nucleus is, now mark my words and be able to use them wisely, okay? Make sure you write this stuff down. It's double membrane. So it has, if this is the nucleus zoomed in, it has one layer of membrane on the inside and one layer of membrane on the outside. So it's a double membrane. And where both these layers meet, you get a gap called a nuclear pore, right? And this pore can be fitted with, remember the cell membrane, we're going to talk about it in more detail, but be able to recall it. If you can recall it, it's amazing, because then you realize that this is two membranes, and where both of them meet, they form a pore, and that pore can be fit, it can be empty, or it can be fitted with a protein, or it can be continuous with, let's say, a channel on the outside, right? So essentially, the nuclear envelope is really tight. Nothing comes inside the nucleus without regulation. Nothing leaves without regulation. And yet we have these small pores. And why do we need these pores? We need them because there's something being made inside the nucleus that needs to leave. And so for that specific thing to leave, you need nuclear pores so that it can escape, so that it's not stuck inside. Otherwise, there's no use, right? And within the nucleus, you can see that there's DNA structure. It's fine threads like these called the DNA. And then there's something called the nucleolus. Now, I need to explain this to you over here. DNA, essentially deoxyribonucleic acid, if you remember, is just phosphate with phosphate backbones with sugar molecules, and then remember those four bases, adenine, thymidine, etc. So all of that combines together to form that, that helical DNA, right? When that DNA is in thread form, it is more open to being used for a process called transcription. Transcription essentially just means conversion of writing or conversion of information from one method to another. For example, if I'm converting my spoken speech into written letters, then that's transcription. If I'm converting my DNA coded information of base pairs into mRNA, which is essentially what happens, that's transcription again, because I'm coding from one language to another language, right? Then what is the nucleolus? The nucleolus on normal staining is a dark blotch against this lighter stained rest of the thread-like network. So if you recall on the top of your head, if you stain a nucleus that is heavily dividing, you see something called chromosome bodies, right? And those chromosome bodies come out darkly stained as well. And why was that? Because they were very tightly packed DNA. They were not loose at all. And essentially the nucleolus is just that. It's a very tightly packed DNA. When DNA is so tightly packed, it becomes really difficult to loosen it up. And hence it is also stained more, right? Let's see its functions. Before looking at the functions, we need to understand what the DNA works like. So follow my pointer here from this area of the diagram. We know we have a DNA which is basically anti-parallel strands which have a backbone, and then there's base pairing in the center. And then how So what's the difference between those terms? Chromosome, chromatin, chromatid, all of those terms keep coming at you sooner or later, and you're always confused. Most people come up to me, they're confused. So let's end that confusion once and for all. So this double helix structure is the DNA, right? When this DNA binds or turns around proteins, it forms a rather concise structure. It keeps turning around proteins, essentially 8 histone proteins joined together for 2.5 turns of DNA to loop around them. And when many of these looped structures join together to form this compact structure of DNA and histones, it's called a chromatin fiber. So forever remember this. It's a chromatin fiber when it's packed up. When many chromatin fibers pack up to form this X-shaped structure that's very famous, it's called a chromosome. It's no longer called a DNA. When that if we talk about only one limb of this chromosome, only this part and not the other one, for example, if I separate this in the center, I'll have a worm-like shape, right? This part, if only one is present, it's called a chromatid. So DNA, chromatin fiber, and then chromosome. If the chromosome exists as only one wing-like structure, let me just give you an example of that. So if it's a structure that's essentially like this, it's called a chromatid. It's called a chromatid with a D, as compared to a chromatin, which is just wrapped around, which is just DNA wrapped around its histone proteins, right? And how does this wrapping occur? Essentially, they're both oppositely charged, which is why they're able to wrap around each other. So let's see what a nucleus can be like, right? This is the general structure. It's a double membrane organ. We've already understood that. There's pores in it and there's structures in it, which are either nucleolus or the DNA itself opened up. Nucleolus is highly packed DNA, just like I told you guys. So over here, before moving on, there's also something important I feel like that should be discussed here. So more details of this specific topic come in when you're doing your mitosis and similar chapters. So you've read that there's 3 structures. So there's one DNA, and when you transcribe stuff, you turn it into an RNA, right? And an RNA can exist in 3 forms. It's either a mRNA, a tRNA, or a rRNA. So I'm writing this here because this is important, right? mRNA is used for making protein. So write that down. mRNA is used for making protein. We'll see how this is a part of your future discussions. tRNA is also used for making proteins. It's that weird clover-like shape that brings the amino acid, and helps at the ribosome. Remember, there's a structure called the ribosome where translation occurs. So that's where the tRNA comes in and fits and brings protein. And this is the mRNA. So all 3 of the RNA are supposed to work together to produce that protein, right? And that sequence of beads made of amino acids that make a protein, all 3 of them are supposed to be together. The mRNA comes from this DNA that's opened up, and we get it over here. The tRNA also comes from the nucleus because it's an RNA, right? Where does the tRNA come from? It comes from this DNA as well. The rRNA, what's the rRNA, right? The rRNA is essentially in this ribosome. So now all 3 of them are coming together, right? The rRNA combines with proteins to come inside this ribosome, where eventually mRNA, tRNA, and rRNA will help in making this protein, right? This rRNA is made in the nucleolus. So the whole point to understand the nucleolus came here, right? So we have that one. We have that now. Okay, so let's move on and see our next topic. Now, we're talking about the nucleus generally, so we know the location. The location is at a side in an animal cell, in the center, in a plant cell. The contents we've talked about. Now let's talk about the shapes and the lobes, which is why we're here essentially, right? To understand what's different from your A-level content. So the shapes and the lobes can be different. So the nucleus essentially can occur in different shapes. It's not necessary that it's round, right? It can be bean shaped, let's say, for example. Or like I want to show you in this diagram, the only thing that stains differently here is the blue part, right? And you must be so confused, like, what is this blue thing right here? It looks like a nucleus, but is it like many nucleus? Is this a cell that's malformed? No, not at all. It's a multi-lobuled, many lobes of one nucleus, right? So it's a multilobulated nucleus, and this is a characteristic nucleus for neutrophils, which are your immune cells, which are your your leukocytes, right? But then while staining, if this nucleus is staining this way, then why, if these are cells as well, why aren't they staining? Which means it does not have a nucleus. So where did that nucleus go? And that's part of our future directions. That's part of our future, process. But right now, we need to understand that it's not necessary that the nucleus that you studied about as a kid will always be the nucleus it is. What you need to understand is the integral structure, what's inside it, what's being made from it. What are those, the three things that are being made? What functions do they perform? So they form proteins, they form, they help in, regulation of stuff, they help in forming, for replicating the cell, right? So you need to understand the integrity of the structure. And even if the structure comes in with a different shape or a different color or not at all in case of these cells, which are the red blood cells essentially. So this is your blood smear. This is what your normal blood looks like, right? And so if you look at the normal blood smear, all your life you've been studying that, okay, all cells have nucleus or they're dead cells. And then this is your normal blood smear where two cells as compared to the 30 behind them have a weird shaped nucleus, and those 30 do not have a nucleus at all. The red blood cells have no nucleus at all, right? So, and at some point in their life, they did to grow up, but then they obliterated it. They deleted their nucleus, and which means essentially it is important to have nucleus for life. But then we know essentially the red blood cells survive nearly 3 months without that nucleus, which means the functions the nucleus performs is more important than the integrity of the structure. It means if it's making correct proteins and storing them correctly, your cell can survive up to 3 months, 120 days, right? And if it's not doing that, your cell will die. So having or not having a nucleus no longer remains a question. And that's how, because you're doing medicine, applying for medicine, this is an important concept to understand, because this is not only going to help you understand question stems. There's going to be questions that come up and they write off as, okay, there's a nucleus, that a cell with no nucleus doing this and this, and you'll be like, a cell with no nucleus, you'll be so confused. So know that is possible. Actually, it is the case with red blood cells, right? And then there's a lobulated or a bean shaped or many different shapes of nuclei. So the essential thing to understand is this. Now, this is a very beautiful diagram. It explains the endoplasmic reticulum. Your book definition always says an endoplasmic reticulum is deposition or is a double membrane organelle stacked against the nucleus or stacked in a corner if it's the Golgi apparatus, right? So let's see what this double membrane is. Now, one thing this diagram shows very beautifully is the double membrane, right? So if this is the nucleus, let's start from here in the diagram. There is an inner membrane and an outer membrane. So there's two nuclear envelopes or nuclear membranes. And these nuclear membranes have pores in them. And these pores are lined by these proteins, right, to keep the pores open. If they're not lined by the protein, so over here, it's not lined by a protein. So it forms a channel to the next structure in it, next to it, right? So this channel or these pores open up to another double membrane-like structure called the endoplasmic reticulum. Endoplasmic reticulum is always next to the nucleus. One thing you need to remind yourself again and again, it's always surrounding the nucleus. If it's not, if it's lying somewhere in a corner, not talking to anyone else, making its own vesicles, making its own stuff, that's the Golgi apparatus, right? So whatever is leaving the nucleus, which we know is the RNA, comes in contact first with the endoplasmic reticulum. And so the endoplasmic reticulum is the site where we will have our formation of protein. So the translation occurs in the endoplasmic reticulum. Now, here it's important to understand that this is this is important to understand that not all cells have an endoplasmic reticulum, right? So plant cells do, animal cells do, what cells don't? So I want you to keep that thought in your mind and we'll get back to that, right? So the endoplasmic reticulum, these two folds do not go to waste, okay? These two folds, this first fold and the second fold, is very useful because the inside is called a cisternal space, right? And this cisternal space is where your protein will be formed on the surface in these yellow dots. The mRNA, tRNA, and rRNA will come and form in these yellow dots, and the proteins will be sent inside so that they're protected and they can be modified. So what are these yellow dots? They're the ribosomes, right? The ribosomes or the structures made from the rRNA. So now you know that one structure for protein synthesis is already present here, the ribosome. Two more structures need to come here, the mRNA and the tRNA. So let's see how we reach them, right? Okay, so essentially, we have two kinds of endoplasmic reticulum. If you look here carefully, right? So we have a form of endoplasmic reticulum that is smooth and another form that is rough. The smooth endoplasmic reticulum is smooth because it does not have ribosomes. The rough is rough because it does have ribosomes. Now, one thing was clear, that ribosomes were necessary for formation of protein. Then how is the smooth endoplasmic reticulum different? So let's see, let's head on to that and see what happens there. So let's see what happens at the reticulum and specifically at the rough endoplasmic reticulum. So a molecule, like I said, a molecule called a ribosome comes in and attaches here to the endoplasmic reticulum. When it comes and attaches, the mRNA, look at this mRNA. The mRNA is a strand of base pairs. It comes and attaches in the mouth of this ribosome. And then different tRNAs carrying different amino acids will come, form their bonds, and form amino acid proteins. These protein chain, where will they end up? They will end up inside the cisternal chain, in the cisternal space, right? And this cisternal space will then protect it and help in modifying it, right? For example, this has a special enzyme inside that is going to attach, let's say, a glucose molecule to the protein. So now this protein will become a glycolipid. So initially this protein was, let's say, an enzyme. Now it's no longer an enzyme. It has a glucose molecule to it. It's, for example, used in making you move faster or something like that, right? So you need to understand that translation, the polypeptide linkage, protein synthesis, all of these are alternate words for the same thing that occurs at this rough endoplasmic reticulum. Not only does the rough endoplasmic reticulum does this, but it also modifies the protein. Like I said, it modifies it from an enzyme structure to a non-enzyme structure, from a non-functional enzyme to a functional enzyme. So all of this is done over here, and eventually this will then be prepared to leave the cell or be used within the cell, right? And then the last point here, sterol synthesis. So this is an important point because you need to understand that the smooth endoplasmic reticulum is also lying outside here. What is this responsible for? This is responsible for synthesis of your fatty acid components, for example, sterol, right? Because the body is either obtaining your fats from the outside or making them yourself, just like proteins, right? So how does it make these fats? It makes it at the smooth endoplasmic reticulum. So that's essentially what the rough endoplasmic and smooth endoplasmic reticulum is. The rough endoplasmic reticulum, you can see here again, right next to the nucleus, double membranes are connected together so that if a protein made here can travel all the way out over here and leave the cell or enter the cell, right? And then it has little ribosomes stuck to it. The smooth does not. Looks beautiful, like the coral reef structure, right? And the smooth is essentially synthesis and storage of lipids. Similarly, rough is also proteins are made but can be temporarily stored as well, right? So that's what essentially this membrane is for. The last membrane before we move on to the next organelles is the Golgi apparatus. Golgi is again double membrane with a cistern in the middle. What's the difference? It's not starting next to the nucleus. It's somewhere alone. It's a stack of membranes being placed alone. How is it different? A Golgi apparatus is responsible for modifying and making that protein functional. So once your protein or your lipid leaves these rough endoplasmic reticulum, it forms these small vesicles, membranes that have bled off. Those membranes eventually come along and join together to this structure where they will deposit all of their material inside. The Golgi will have enzymes and other things inside that are, or for example, vitamins inside that are useful because, for example, if you want to bind your vitamin A with the structure in a protein to make a vitamin A protein, your Golgi will do that. It will modify, it will pack, it will make sure that it's functional, it will make sure that it's not a waste of your protein or your energy, right? The structure of the Golgi, very easy, keeps coming up in the books again and again. There's a cis and a trans phase. And the trans phase is the mature phase. You can see all of that structure that was entering here from the endoplasmic reticulums. So where does the Golgi get its supply? The endoplasmic reticulums, right? It's being modified in the center, in the lumen or the cisternal space. And then eventually in the trans phase, it forms these vesicles. It's packaged into vesicles, which are again, vesicles are essentially now their single membrane, right? The blebbing off from one side of the membrane to form these vesicles that have mature protein in them. Now this can be secreted, called the secretory vesicle, or can be used within the cell. So that's essentially what the Golgi body or the Golgi apparatus is.